My name is Guillermo Herrera. I am the chairman of pathology at the Department of Pathology at the University of South Alabama in Mobile, Alabama, United States. I've also been very interested in research, and my main area of research is that of the interaction of monoclonal light chains with the glomerulus, more specifically with mesangial cells. The beginning of amyloid fibril formation occurs in the mesangium, and so the interactions between the monoclonal light chains and the mesangial cells are crucial for the process to occur. There is a receptor on the mesangial cells that we discovered approximately 6 years ago, which is called sortilin, goes by the name of SORL1. And this sortilin receptor is where the monoclonal light chains interact with the mesangial cells, and they get sorted. That's why it's called sortilin. And depending to what type of monoclonal light chain, what kind of sorting will take place. In the case of those light chains that actually are engaged in the formation of amyloid fibrils, the light chain get endocytosed into the mesangial cell and transported into the endolysosomal compartment, where the lysosomal enzymes act on the light chains and eventually form the amyloid fibrils. My colleague, Dr. Elba Turbat-Herrera, myself and my team have been working on mechanisms of glomerular damage in monoclonal light chains for more than 30 years, and we have slowly but surely come to an understanding of how this process occurs. Elba, you may want to say a few things about this process.
Well, I was just going to say that we have always been studying the process of amyloid formation and where it happens and the damage it causes, and also we're now engaged into the repair. Guillermo, you want to tell him something about it?
So what we presented in this meeting, one of the presentations was to actually visualize with transmission electron microscopy how the interaction of monoclonal light chains with lysosomes occurs, and how the early formation of fibrils occur in the lysosomes.
Correct.
Actually, this you culture the human mesangial cells, and you incubate these human mesangial cells with light chains that we purify from the urine of patients with light chain proven AL amyloidosis. And when we incubate these cells engage in the formation of amyloid. And what we illustrated in this meeting was how this formation can occur so early. It's about 2 hours from the time we put the light chain in the culture media that the mesangial cells are able to form this amyloid fibrils. And so the process of formation and how the amyloid fibrils get extruded into the extracellular matrix where they eventually reside, that's where the fibrils in amyloid occur. It's a process that now we understand in detail. So this gives us an opportunity to work on therapeutics to avoid the damage in the kidney in these patients who have this disease.
But maybe you should tell them how the mesenchymal stem cells from the bone marrow have been used by us to try to repair the damage that the amyloid causes. And that is a beautiful work. You can actually see the cells eating the debris. You can actually see the cells eating the debris and destroying it. And that is really amazing.
So the mesenchymal stem cells are cells that the first thing they do is they find the site where the damage has occurred in the kidney. They migrate to that area and they clean the environment, so they actually phagocytose the amyloid fibrils and destroy them, cleaning the environment. Then those same mesenchymal stem cells transform into mesangial cells functionally and morphologically.
Correct.
And then they rebuild the mesangium and put it back together. So you can see the importance for a patient with AL amyloidosis and kidney damage, how you can come back and repair the damage that the light chains have done. And of course, the treatment will include treating the plasma cell clone to avoid more light chain production and therefore more damage in the kidney. So you stop that and then you rebuild the damage that occurred, you have a brand new kidney to work with, which is the object of this research.
And like I said, we use the mesenchymal stem cells from the bone marrow, not from embryos, which we don't agree with, and the bone marrow works just as well. So it is wonderful, and when you see the mesangium the area has been cleaned and repaired, it looks like normal.
And keep in mind that this research has been slow. It has been a research for the last 35 years, and that's what research is. It just takes a while to figure out all the steps involved in a given process, and that's what we have been engaged in. So we may have about 20 or maybe 30 different publications regarding how this research has evolved over the years, and we're very happy to say that today we understand how amyloid formation occurs in the kidney, in the mesangium, in patients with AL amyloidosis.
Yeah, and we have also used scanning electron microscopy to prove this, to see how the cells actually produce amyloid, and it is amazing if you would see these images.
So in scanning electron microscopy, what you're looking at is the surface of the cells. So it was a perfect methodology to look at how the light chains interact with the surface of the mesangial cells in producing damage in the mesangial cells. And it's also a great way to visualize how the amyloid once formed in the lysosomes get extruded by the lysosomes going to the cell membrane and actually letting the amyloid fibrils escape into the extracellular matrix. Those changes that are really surface changes can be very nicely visualized with a scanning electron microscopy.
And what we do is actually a translational research, which means you discover it in the laboratory and translate it to the patient, to patient care, because that's the ultimate desire.
More recently, we have been working on tubular injury in these patients, which also occurs, and trying to figure out what the mechanisms are in the tubules for producing damage, which is a little different than what happens in AL amyloidosis.